Today I will share a small overview about making of protein from DNA. At first, what is cell? Cell is a base unit of every living tissue. In most of the human tissue, there is a human cell. There is a structure called nucleus. Nucleus contains genome. What is genome? Genome code. We'll discuss about this topics later. So genome. So genome is split between 23 pairs of chromosomes. Each chromosome is a long strand of DNA. Okay, and packaged around a protein. Long strand of DNA that tightly packaged around a protein called histone. Within the DNA there is a gene. Gene contains instruction for making a protein. It given by Allah subhanahu wa ta'ala, it's automatic, this instruction. Then when a gene is switched on, there is a specific enzyme, RNA polymerase is activated. Specific, not a second one in second instance. So when a RNA gene is switched on, an enzyme RNA polymerase comes up and attaches to the start of the gene. Then it moves along with the DNA until the making of strand of messenger RNA. So it moves along with the DNA, making the strand of messenger RNA, and it done out of free basis without any effort. So it is done by Allah subhanahu wa ta'ala. So it happens to in the nucleus. Then DNA has a coding power. What DNA after that does is DNA code determines the order in which the DNA determines the order in which the free bases are added to the order. DNA determine that order in which the free bases added to the messenger RNA. Then that process is called transcription. After the transcription period, that messenger RNA cannot be used for producing the protein. So there it should be processed. It cannot be used as a template for producing the protein. So it needs to be processed. So for it to be processed, there is a removal and adding part in a messenger RNA, that is intron, exon. We'll discuss about these topics later. So the messenger RNA then comes up or moves out of the DNA, moves out of the nucleus, okay, and goes into the cytoplasm. And we know that there is a ribosome, protein factory in the cytoplasm, that is called ribosome. So ribosome comes and binds with the messenger RNA, and ribosome has a reading power. What it reads is ribosome actually reads the messenger RNA. Okay, so ribosome comes up and reads the messenger RNA. Okay, reads the messenger RNA, reads the code of the messenger RNA to produce a chain of amino acid. Now, amino acid is carried out by the transfer RNA. Specific transfer RNA carries a specific specific amino acid. So transfer RNA carries that amino acid. There are 20 types of amino acid in our body. They carry that amino acid into the to the ribosome. So the messenger RNA is read 3 bases at a time. As triplet is read, the transfer RNA delivers the specific amino acid. One by one transfer RNA comes, and one by one transfer RNA comes, and codon anticodon part is being all the step is being done. Then it's delivering continuously its own specific amino acid. The first transfer RNA done comes, and its codon part, anticodon part is fixed its a specific codon. Then it goes out and it delivers its own amino acid. So one by one this amino acid is released and forming a chain, released and added to a chain of amino acid continuously by the peptide bond. A polypeptide chain of amino acid is formed. So until the last after the addition of last amino acid to the chain, suddenly that chain turns up or shapes itself as a 3D. It turns as a 3D dimensional to form a protein. It's needed to folds into a complex 3D shape to form the protein. So then it forms the protein.